Spatial biology is changing the way tissue biopsies been looking at for the past century. 5 years ago, we couldn't even imagine that we can do something like this, that we can one day measure millions of cells in a small piece of tissue, the vast amount of information that by human eyes we cannot see. Now we get millions of measurements that allow us to understand the complex disease from the patient tissue.
We're in a very unique position here at QIMR Berghofer. Our spatial cell biology facilities cover everything all the way from simple slide scanning to being able to detect hundreds of proteins and thousands of genes in patient samples. So these instruments allow us to create very detailed molecular profiles of individual samples and patients.
What we are seeing in this image in here is the H&E image. This is the standard of care image that pathologists look at routinely to tell about the disease. As you can see, there are many two colors, blue and pink. In here, this is a spatial slide where we can measure hundreds to thousands of molecules showing at different colors here. To generate the spatial data, it takes time and it costs money. To bring spatial data to pathologists to help with their daily diagnosis, we developed a machine learning algorithm that can take H&E images as the input to predict the spatial information without adding more workload burden or cost to the pathologist. Early stage cancer, they still look very similar to healthy cells, but inside them, among millions of biomarkers, 2 or 3 of the biomarkers are showing the differences. And our technology allows us to pick up those 2 or 3 early stage cancer biomarkers. The spatial technology and the machine learning tool together are giving the pathologists a super resolution eyes so that they can make decisions faster and more accurately. It's almost like Superman or Superwoman eyes that the pathologists now have to combine with their very own eyes and make a better decision for their diagnosis. The results are not from AI, they are from pathologists who are well informed by their own eyes, and so the patient can really trust the result. Building an AI model is a task that requires the combination of mathematics, of good data input, and a strategy on how we build the model. Now that we have proven that STimage works for breast cancer, skin cancer, kidney cancer, and liver disease, we need to expand the application and training of STimages to other cancer types and other disease types as well. Another benefit of the tool is to help people in remote or regional area to have a better access to pathology diagnosis.
It definitely does feel like spatial has the potential to be that revolutionary instance in history where if we get this right, it can really explode and propel science in an upward direction that we couldn't imagine.
Cancer is so difficult to deal with, and it needs new technologies, new knowledge to advance the field. So that really motivates me every day to work on this. I'm so excited and very hopeful too that in the very near future, what we have achieved so far can be applied and benefit patients.